I really thank the organizers, Dr. Shomik Ghosh and dear chairpersons to be putting me here. And my talk is molecular diagnosis in pulmonology. So when we are talking of a pulmonary phenotype, we actually probably have, it's like a triangle which we are talking about. There are developmental issues, there can be environmental issues and genetic issues, which can define the etiology of this particular phenotype. I would be concentrating on two features, the environment and the genetics. When I'm talking about genetics, because it's something which is very close to me, I'll take this up as first, but though this condition would come up as rare. When we are looking at genetics, there are different aspects that we are looking at. Initially, before this technology expansion and before Human Genome Project, before we could sequence the human genome, it was more dependent on what symptoms are there and what biochemistry we could follow. But now, because we can interrogate each cell, we came to know if there is a structural or numerical problem which we are looking at, or whether we are also looking at single genes or a set of candidate genes. Now, because the technology is so that we can interrogate each base, right now, the genetics of pulmonology diseases, pulmonary diseases, are expanding a lot. And what can we do with this technology? We can actually have an early disease identification. A few talks back, Dr. Indu actually highlighted how early disease identification could be helpful. We could go for precision medication, and also we can have an idea about the disorders to prevent it in the next generation. So if we are looking at it, this was what she had presented. I'll just summarize it once again. Can I have a pointer, please? Can I have a pointer? So if we look at it, we have here the structure of the lower airway and the alveolus, and what we know, I shouldn't be talking about this in this forum, but we know that, there are these cells that the type 1 and type 2 cells which are lining up, and they are actually, they have these normal lamellar bodies which would be secreting the surfactants. Now, any problems in these surfactants, which is related to SFTPC, will actually cause milder disease, because here this lamellar structure remains as sort of round, but it's not that disorganized, but normally mutations which are there in the SFTPB or ABCA3 mutations, they cause some disorganizations resulting in more severe kind of diseases. What we shouldn't forget is also the pulmonary alveolar genes which are there. Normally mutations in these genes do not cause any problem in the structure or the function, but what it causes is in the homeostasis, which interferes with the anti-GM-CSF antibodies. Also, we shouldn't forget the neuroendocrine cells, which actually affect the lower airways, and all these are our surfactant disorders, which will present in the neonatal period or in the pediatric period. The other common genetic disorders which we have are related to the bronchiectasis, and here very commonly we have the primary ciliary dyskinesis, which has over 40 genes. Obviously cystic fibrosis is abnormal chloride transport or abnormal sodium transport, or the primary immunodeficiency genes, which is 200 plus genes. Earlier, the only diagnosis probably we would have done was just CFTR and looking for 5-6 mutations. So just from these slides, you can see how the expansion has happened, just because we can interrogate the basis. Also, there are diseases which we know which are not really diseases of the lungs, but they do have an impact in the lungs. Like when we are looking at Marfan syndrome, or we are looking at a Wilson disease, or an MPS, or a Cornelia de Lange. So the disease expansion has happened a lot, and depending on the type of mutations, we are actually able to also understand what to treat and what to refer, and maybe improve the quality of life of these patients. Now, if we are looking at the diagnostic platter, we have a lot to offer. As I said, we can now interrogate the basis. So we have shifted from the traditional karyotype to the microarrays and the MLPAs and the genome sequencing. When we are looking at genome sequencing, if we are looking at a smaller panel, like say I'm just looking at the primary ciliary dyskinesis, I might just go for a gene panel. Or if I'm looking for maybe a PID, like a primary immunodeficiency, I will go for an expanded panel like whole exome sequencing. Now, what test to do would many a times this is where we are stuck. This would depend on what genes we are interrogating. Now, if I'm looking at maybe a gene which is related to pulmonary alveolar proteinosis or surfactant disorders, we know they are single gene disorders and there is single point mutations. So, an exome sequencing is probably something which I will prefer. But maybe I am also looking at a gene like CFTR, which has 90% point mutations, but 10% are deletions and duplications, so one test might not suffice. I might need to go for another test, which might be the MLPM. But if I'm looking at a gene like SMN, or I have my diagnosis as 22q deletion syndrome, the exome sequencing might not be useful, or it might be too much, and we have, for SMA, we have to do an MLPA. So what I'm looking at, my clinical diagnosis will lead to what exactly genetic test I want to do. Also, sometimes the phenotypes are overlapping. When I say the phenotypes are overlapping, it might mean that this child has respiratory syndrome, this child has intellectual disabilities, the child has many other things. Now, if we don't jot it down, we probably might miss a gene. If we are just focused on one organ, we might be missing a particular gene or a particular disorder. The reason being that each of the phenotypes you write are the HPO terms, and each of these terms have a list of genes that can be associated. So when you send a test, what the lab does is for each of these HPO terms, the gene list is basically enriched, and that would be the list they would be looking into for your patient. And that will help them to get a number of variants. Now, these variants which you get, there are thousands of these variants, which will be filtered on basis of quality, and then on basis of your case, and also on basis of the inheritance pattern or the pedigree which you are noting. On basis of that, you will get your report. So if you are just writing respiratory distress and not actually explaining any other test or any other form, then you might get a report which is, not sufficient or not enough. But if you are writing a detailed phenotype, you are writing a detailed case history, you might just come up to a gene which is there. If you're writing less, you only get the low-hanging fruits. So now the sequence is done, the report is there. It's mostly, a section of ATGCs and mutations in it. So what does it mean? So what it means would depend on what we are actually looking at. So when we are looking at a report, we will get 4 kinds of classifications. One is pathogenic, likely pathogenic, variant of uncertain significance, or benign. The pathogenic or likely pathogenic is something which we don't really worry about, but if we are looking at VOUSs, that is something which we are very worried about. It's like, somebody has just put us on a hold, and we don't know what this is because it has limited scientific evidence. But how do we go about it? So this is a case where we have 2 CFTR mutations in a child. Now, if it is 2, we know that CFTR is both autosomal dominant and autosomal recessive. Now, if it is autosomal recessive, then it has to be in the trans. By that, what I mean? I mean that we have to test the parents and see if both the mutations are coming from one partner or the 2 mutations are each coming from each partners. Only if it is in trans, we can call it as cystic fibrosis. Otherwise, it will have milder symptoms and the implications are different. So just a report means nothing. We also need to look at the parents, and we also need to understand what variants we are looking at. Now, see here we have a likely pathogenic variant, which means that this is a reported one, and we have a variant of uncertain significance. Now, as I said, if it's uncertain, then we shouldn't throw it off, but we need to investigate if it is clinically relevant, if it is matching our phenotype. Now, this is another case. This is a ABCA3 surfactant metabolism, like mutation. And here you can see we have 2 uncertain significance genes. So obviously my first thing would be these are autosomal recessive disorders. So I will see the segregation pattern in the parents. But if it's a VOUS, can I reclassify this? We can do that. If we do that, we have to follow certain guidelines. So first we see whether it's matching the phenotype. If it is matching the phenotype, there are databases like OMIM from where we can look at it. Also understand that genes right now, earlier we used to see CFTR, say cystic fibrosis, but right now every gene has an expanding phenotype. So there are apps like the panel app, and there are apps where you can actually look at what these genes are implicated in. Your patient might have a new phenotype which might not have been described, so we should be very careful to what we are seeing, and if this was reported elsewhere to any of these expanding phenotypes. Now, once you have established that, then you can go for the ACMG classification. When we are going for an ACMG classification, then we can reclassify. The ACMG is divided into 4 sections, the classification part. One is very strong, then strong, moderate, and supporting. So if we can actually have these pointers, then we can reclassify an uncertain significance variant into a likely pathogenic or even a pathogenic one. So variant interpretation has its own chain of thoughts, and this should be done, and this is what is done by a geneticist or the lab at the back end to reclassify the report and reevaluate the report if needed. So just a test will not matter. So now we have a diagnosis. Can we know more? Now, that is where the personal or the precision medicine comes in. So in this case, what we have, this is an example of cystic fibrosis. Now, the mutation can have a change where the protein is not synthesized, or the protein is misfolded, or the protein has certain other defects. Now, depending on what kind of defect it is, we will see the severity of the disease. But just let's put some cricketing into this, that see, none of the genes are working alone. It's always working in a team. It's a system, so it's not alone. So every phenotype will also have modifiers. Now, these modifiers can be positive modifiers, and these modifiers can be negative modifiers. Just like in the game, we do have a man of the match, but this man alone cannot play the entire match. What we need is a team. And even the genes, even though there is a candidate gene and there is a significant gene, we need the other modifiers to generate a phenotype. Why is this important? This is important because in that case, I can actually give, if I have the mutation, whether it's a single mutation or a double mutation, then I have a modifier. This modifier will help me understand in a disease which is as complex as cystic fibrosis, that which type my phenotype is, whether it's severe, whether it's less severe, and how will the phenotype modify. So my phenotype will actually depend on my type of mutation, the number of mutation, and also the modifier function. For diseases like cystic fibrosis, this is extremely important to understand the progression of the disease. Now, why is it important? Because CFTR right now has drugs. And these drugs are available, and each of these drugs are based on the function. I'll not go into the details because I am just 2 minutes short of my entire presentation. But as I said, like how the protein is, whether it's modified, whether it's no protein is being produced, or how it is changed, will give me what drug I am using. So there are ways right now, there is the sites where you can put in these mutations and see what choice of drugs you are getting. Yes, these drugs are very expensive at the moment, but we do have the rare disease policy, and the rare disease policy are supporting these drugs. So if your patients are registered at the Center of Excellence for all these rare diseases, they can get the drugs and you can give them a choice. So CFTR is just not the only therapeutic target in lungs. There are other diseases where such therapeutic targets are there. And as I said, as the phenotypes are expanding, so are these molecules, and so are the therapeutic targets. Now, this is another one. This is an SMA patient. This is a patient which was jointly looked with Dr. Sanjukta De and me at Peerless Hospital. And here, this patient, we detected them very early with SMN. And fortunately, this patient got the drug, the 16 crore drug, and the child has gained all the milestones and is doing very well at the moment. And the drug she got was nusinersen. So, even in a cost-restrictive setup, everything is not impossible. Now, initially, the thought is to do genetics when you have ruled out everything out. But, we are moving to a place where that might not be true. If we do genetics before, that might help us in diagnosis. Like in India, there are a lot of deaths below 5 years, and many of these deaths are undefined. And if we have these, we can classify these by newborn screening or newborn genetic sequencing, then I need 5 more minutes. So that helps us in early detection, and early detection can actually help us in early diagnosis, prevention, and better management. Now, if we are looking at the environmental causes, this is where I would introduce metagenomics from Metagenome. So Metagenome has these series of tests which are actually looking into the infectious part of it. Now, this is more common than what I was speaking before. Now, if we are looking at it, what is metagenomics? Metagenomics is basically what we are looking at when we are looking at the microbes. Now, these microbes can be from the source can be anything. But now, if we understand the entire genome of this, then what happens is that we understand the organism, and probably we will also understand the treatment plan. So if we are looking at it, why can we understand this? So every virus or every bacteria, all these microbes will have a conserved region. And these conserved regions actually place them into the phylogeny or the taxonomic groups, so that we know this organism is closer to the known organisms which we have, and hence we know the treatment plan even if it's a new organism. So these are the clusters that we can put them into when we are looking into it. So metagenomics is basically looking into sequencing, like sequencing the entire like the sample and looking at what microbes are there. So normally you have to culture, but normally you have to culture. When we are culturing, then there can be culture failures. There can also be selective selection where the microbe of interest is left out. But when you are sequencing, you actually get the entire picture. So when you get the entire picture, so you can distinguish the viruses and the bacteria, you have the pathogenic profile, and you also can identify novel pathogens, and there is accurate detection also. So if we are looking at it, there can be 2 ways. One is targeted. When in targeted, we have these targeted panels where we are looking at the conserved region of these viruses or bacteria and giving you the report, but these are mainly the known ones. But metagenomics helps us to explore the newer ones and also helps us to give us a better variety of these reports. So there are recommendations which right now are being made. And if we are looking at it, the reports will look something like this, like in NGS space, like what is the percentage of species we are getting. And at the same time, it will also give us what is the related species we are getting. When we say OTU, these are operational taxonomic units. So each of these viral or bacterial genomes, how closer they are and how closely associated they are. In this case, if you look at it, you see here in this particular, in the third row, you will see that in the control it's 0, and in the patient it is 11%. So this might be the one of significance, and a treatment plan by this plan will be helpful. So also Metagenome has this SPIT test, which actually is looking at SPIT sequencing, which is actually looking at the Mycobacterium tuberculosis, and the sensitivity of these tests are 100%, specificity is 98%. And if we are looking at it, they have published this data where in the sequencing has helped to understand what type of resistant mycobacterium we are looking at it, and it's beneficial when we are looking at a XDR strain. Now, when we are looking at an XDR strain, there are multiple drugs that we need to look at, and if we are sequencing it in one go, we can actually give you sort of a hint towards what drugs can be used, because the pharmacogenomics is also coming up. So the pre-XDR case patients and the new drug regimes can be actually understood through the SPIT-seq sequencing. I'll skip this slide, but I'll keep this one, that today is probably, what we do is probably hypothesis driven, that we understand that something is there and then we are trying to investigate. But probably what we will be there tomorrow is something where it's data driven. So we do the sequence, we understand what it is, we treat it even before the symptoms comes, so that we have lesser patients and more pre-symptomatic asthmatic patients, and we can give them a better quality of life. Thank you.
Thank you, Dr. Dutta, for speaking on a very difficult subject in a very simple way. Thank you.
Thank you.